Okay, so let's start. I think it's 11 o'clock. Welcome to the Diversity in Biology Seminar Series event. My name is Anders Gleggers. I'm a professor at the Department of Biology. And it will be my great pleasure to introduce today's speaker. But before I do that, I have to and would like to acknowledge funding for this event, which is provided by the UBC Anti-Racism Initiative Fund, the Irving K. Barber Faculty of Science, and the Department of Biology in UBCO. I would also like to acknowledge that UBC Okanagan is located on the ancestral and unceded territories of the Silix Okanagan Nation. And it is likely that many of you are joining us from Kelowna and other parts of these territories. However, many of you, probably including our speaker for sure, are joining us from other regions. So I would encourage you to reflect on the lands on which you live and work and who the ancestral caretakers of these lands might be. So now, as I promised, it's my pleasure to introduce Dr. Villanueva, who is a Filipino-Canadian scientist. She is originally from Coquitlam in BC, just like 350 kilometers west from here. But she started her scientific career in Waterloo, University of Waterloo in Ontario, where she earned her Bachelor of Science. And then we were happy to have her and fortunate at UBC Okanagan, where Erica completed her master's degree in our laboratory. After that, she returned to Lower Mainland and joined laboratory of Dr. Hayden, working on Huntington's disease at the Center for Medical Medicine and Therapeutics. After that, she moved to Denmark and she completed and defended her PhD in the laboratory at the Biotech Research and Innovation Center of the University of Copenhagen. This is where I last actually met her. And we watched a soccer game between Iceland and France. Next to one of the canals of Copenhagen. And now, well, as I said, she successfully defended her PhD just several months ago. And now she's a postdoctoral research fellow in the molecular pharmacology group at the Department of Drug Design and Pharmacology, and still at University of Copenhagen. And she's joining us from Copenhagen, and it's not too late. It's evening for Erica, so thank you so much for spending your evening with us. The Zoom platform is all yours.
Oh, thanks so much for that awesome introduction, Anders. It's great to be among familiar faces, but also some new faces for sure. I very much appreciate this opportunity to share my research, particularly from my doctoral thesis. And I just wanted to reiterate a little bit of what Anders mentioned in my introduction, but just give me a second here. Of course, my cursor is not working, and this exact same thing happened at my previous presentation, of course, but let me just see if I can't get it working. In any case, the title of my talk today is Type 1 Interferon Signaling in CNS Homeostasis and Parkinson-like Dementia. But in reality, it's just, it's going to be a discussion on mechanisms of immunity in the brain, which are becoming more and more apparent to be involved in neurodegenerative disorders like Parkinson's disease. So with that, but quickly, as mentioned, I'm a first-generation Filipino-Canadian and proudly at that. And my hometown was Coquitlam, BC, which is the unceded and ancestral territory of the Coquitlam First Nation. And my previous 14 years have been spent in neuroscience research, which saying that out loud now is quite a long time. So as Andis mentioned, I actually I completed my master's degree at UBCO, and I was looking at the impact of secreted inflammatory factors from these cells called glia in the brain. And we were looking at whether or not they actually or their impact anyway on neuronal health using cell-based models. Then I moved on to Vancouver, and in the Hayden lab, I used at least preclinical mouse models of Huntington's disease to actually evaluate some potential therapeutics for that. Then I moved to Denmark and joined Dr. Shure Zazde-Navikas at University of Copenhagen to conduct my doctoral research, and that involved looking at neuronal immune gene signaling in neurodegeneration that appeared to resemble Parkinson's disease, but also it appears to be involved. So I'm quite excited to talk about that today. But currently, I'm actually a postdoc in Dr. Hans Brauner Osborne's lab, and I'm looking at the potential role of a recently deorphanized G protein coupled receptor in a disease called multiple sclerosis, which also involves neuroinflammation and neuronal death. So with that, I just wanted to share this because the common theme here is neuroinflammation and neurodegeneration, and this will be the topic of the day. So briefly, I'll have an introduction on what neuronal homeostasis is. Then I'll talk about immunity in the central nervous system, or the CNS, and neuronal immune signaling, which is a topic that is not really well known, because now we're thinking about glial cells and their involvement in immunity, but my focus was slightly different. Then I want to talk about these molecules called interferons, specifically interferon beta and its receptor IFNAR1. And perhaps it's been heard of in the context of COVID, because interferon beta is actually an antiviral cytokine, and I'll talk more about that in a second. Then I'll talk about Parkinson's disease and Parkinson's disease with dementia, and the involvement of defective neuroimmune signaling in this disease. Then I'll move on to what my doctoral thesis focused on, as well as findings and conclusions from that, and also future perspectives. And then, of course, I want to acknowledge all the people involved in this work. So to start, how do we begin to understand a process called, or like, neurodegeneration? But the word neurodegeneration actually does mean death of neurons. And neurons are cells that make up the functional aspect of our central nervous system, which includes the brain and spinal cord. So neuronal homeostasis involves many different factors. It's much like a ecosystem, so to speak. It involves balance of signaling between different cells, and also nutrients and factors that allow these cells to survive and communicate properly. So the major questions are, what keeps neurons healthy and what makes them ill? And these may seem simple, but it's actually quite complex once you get down to the molecular aspects of all these processes. So by beginning to understand how neurons maintain themselves, how the cells around neurons also contribute to this maintenance, maybe could give us an idea of the beginning of what neurodegeneration is. And by understanding that, then we can move on to designing disease-modifying drugs, or at least figuring out targets that would most likely prevent or reverse the process. So neurons control thought and movement, basically. Neurons this is a very simplified version of a neuron, but they basically have a cell body here called the soma, and typically have projections called axons that end here, called axon terminals. And neurons signal to one another via these action potentials. And here you can see two neurons side by side, but in this box here is an area called the synapse. Now, neurons signal to one another using molecules called neurotransmitters, which are released at the axon terminals. A presynaptic neuron, which is on the side where it's being released, and a postsynaptic neuron, which is the term that you would use for the neuron receiving a signal. And the continuation of that signal or the inhibition of it depends on what receptors are actually expressed at the postsynaptic surface. And this regulation actually determines whether or not, or there are a lot of factors rather that determine what kind of receptors are expressed at this surface. So, sorry. Neurons rely on several processes to aid in their function and survival. Now, this is a picture of a brain, a human brain, and the area in the red is called the substantia nigra, which has a very thick population of neurons that produce a molecule called dopamine. So we call them dopaminergic neurons. Now, these neurons actually have their cell bodies here, but they have long projecting axons that that communicate with neurons in an area of the brain called the striatum, which is right here. And now, at a cellular level, this distance is quite far. I can't imagine the exact metric unit of it, but it could be the equivalent of, let's say, stretching one of your legs further down the street, maybe a few blocks away. Now, neurons with that distance require a lot of tight maintenance of how they relay the molecules from their cell body to the axon terminals. And of course, they require a lot of energy to do this. So let me just try and switch the slide. Wow, okay, I'm really not having so much luck here. There. Okay, so there are some crucial processes for this function and survival of neurons. This includes efficient trafficking of proteins and what we call organelles, including mitochondria, that are these pink things here. These organelles actually travel along microtubules down along the axon towards the axon terminal. And this back and forth is quite important for the neuron to be able to communicate properly. What is also important is that the neuron is able to digest and get rid of organelles that are not functioning properly, or molecules that it no longer needs, including excess neurotransmitters. And these processes are through what we call autophagy or mitophagy. And this is important so that the neuron doesn't have this buildup of waste. So I don't exactly remember which cell or which cities have had strikes from garbage collection or whatnot, but you can imagine if garbage pickup did not occur in an efficient manner in a city, the city would grind to a standstill, or at least it would not be pleasant to live there. And it's the same with neurons. They need to be able to get rid of their waste somehow so that they can perform properly. And finally, there's a process called mitochondrial homeostasis, or the health and happiness of these organelles called mitochondria. Now, this is very important because, of course, neurons are requiring a lot of energy to be able to communicate through long distances and also efficiently and fast. So mitochondria are key in providing that energy, also known as ATP. And they are actually also involved in the production of specific neurotransmitters such as glutamate and GABA. So in essence, they not only provide energy, but they also regulate what kind of neurotransmitters are available for the neuron to communicate with. So this requires that they are able to localize at the synapses and, of course, travel back and forth between that soma. So if that can't happen, then the neuron's in trouble. Neurons not only interact with each other, but also with their surrounding glial cells, or these are non-neuronal brain cells. And I'm not sure why this keeps preventing me from clicking, but there. No, still not happening. Okay. Click. Good. So There are three major types of glial cells. First, there are oligodendrocytes, which actually generate what we call a myelin sheath that insulate these neuronal axons and actually allow more efficient signal transduction. So along those long distances, you could potentiate the signal further with these myelin sheaths. Then you have astrocytes, which are these star-shaped cells that constitute, at least what we know, the largest glial cell population in the brain. And also microglia, which their primary role is surveilling the brain for any sort of pathogens or damaged cell debris that need to be taken up. And so my thesis actually focused on these two immune cell types because both astrocytes and microglia have known immunological properties. So specifically, I wanted to look at astrocytes because they are key in supporting neuronal function. Sorry. I'm sorry, I'm having some technical issues here, but there we go. Okay. So astrocytes are actually quite distributed well throughout the central nervous system, and they interact with neurons at synapses, but also at this area called the blood-brain barrier, where they have these end feet that contact the blood vessel that allow the exchange of ions and energy substrates between the blood circulation. Of course, when you eat, that sugar and energy needs to get to your brain somehow, and astrocytes facilitate that. Hey, click. There we go. And astrocytes also support neurons with the production of neurotransmitters and again, energy substrates from what is gained from the blood circulation. But also they're important in recycling excess neurotransmitters, including glutamate, to prevent what we call excitotoxicity when the neuron is actually overexcited or overstimulated. It can actually lead to neuronal death. So astrocytes are key in preventing that from happening. Now, as supportive as astrocytes are, of course, too much of a good thing can lead to bad outcomes. So astrocytes can also adopt what is called a reactive phenotype in response to pathological conditions or damage to the brain. And this can also contribute to the inflammatory process by, they can also generate, for example, pro-inflammatory cytokines or signals that allow that inflammation to snowball. Microglia are also immunologically involved in the CNS, but they also play key roles in neuronal development and also synaptic plasticity. But in basic terms, it means memory formation or learning processes. So without microglia, it's a bit difficult for the neurons to rearrange themselves to facilitate memory formation, and that makes them key. Microglia also function as innate immune cells in adult brain or the developed brain. And they do that by wandering the CNS and phagocytosing, or rather eating debris and any sort of foreign pathogens it may encounter, like viruses or bacteria. So they're responsible for protecting the brain from these sort of invaders. They can definitely propagate an immune response by releasing these pro-inflammatory cytokines, which means that they recruit other mitochondria or microglia to come and participate in this inflammation to try and resolve the damage or the infection as locally as possible. But of course, without control of this process, this can actually lead to damage in the surrounding areas that are actually healthy. And this is one of the theories behind neurodegenerative processes. And yes, of course, not only the pro-inflammatory response could contribute to neuronal death, but also if they are defective, it could lead to the inefficient clearance of buildup of protein aggregates and that can also contribute to activating other microglia and propagating the immune response in that respect. So neurons, though astrocytes and microglia are typically known to be the ones communicating with immune cytokines, neurons have also been found to be able to communicate with these same immune cytokines and receptors. Sorry. Yeah. And neuronal immune signaling actually also appears to play a role in synaptic plasticity and axon guidance. And again, in other words, memory and learning. So what seems to be the case is that the cytokine or immune signaling can have different effects depending on whether or not neurons or glia are actually expressing them. So the origin of these cytokines as well as the context seem to make a huge difference in the effect. Now, I wanted to introduce a personal example of why context and origin matter for communication. Think of it as a cytokine can be like a word. And a word can mean different things depending on context and origin. For example, the Filipino word for rice is kanin. And I love rice. I love eating rice. It's delicious. But when I came to Denmark, I actually learned that the same word, spelled the exact same way, said a little bit differently, actually means rabbit. Now, for those of you who know me, I love rabbits too, just not for eating. So, I mean, it's a very simple example, but it means that the context in which that word is being used and also the origin, like, for example, whether or not you're in the Philippines or whether you're in Denmark, actually matters in how it's being interpreted. And this is the same for brain communication. So with cytokines, if it's, for example, the cytokine tumor necrosis factor alpha or TNF alpha, which I'll talk about again later, if it's expressed by glial cells, it actually affects neuronal synapse formation and can change the way that a neuron responds. But if that TNF alpha is actually expressed by neurons, it doesn't have the same effect, and that function is actually currently unknown. The same goes for neuronal derived cytokines. For example, tissue growth factor beta and interferon beta are typically known as immune cytokines, but when expressed by neurons, but not astrocytes, they have the ability to convert immune cells that are of a pathogenic function to cells that actually serve to resolve that inflammation. So they can switch the function of the immune cell, but only from a neuronal origin. So this is quite interesting. Now, my thesis also involved the investigation of a specific immune cytokine called interferon beta and its receptor IFNAR, and I just wanted to give a brief introduction to that. Interferon beta is actually an antiviral cytokine with many different immunoregulatory effects, again, depending on context and origin. And it signals through the interferon alpha beta receptor, mouthful, but IFNAR for short. And this IFNAR receptor is the common type 1 interferon receptor because there's interferon beta, but there's also interferon alpha, interferon omega, and so forth. IFNAR is composed of two subunits, IFNAR1 and IFNAR2, but experimental studies have shown that interferon beta is reliant on binding to IFNAR1 for that signaling to occur. Now, it's interesting to note that IFNAR is expressed by all cells, including neurons. So this actually could contribute to the differential effects of interferon beta in the body, depending on where it's expressed, of course, how much of it is expressed, and which cells are actually receiving that signal. And this is the basis of my investigation. So there was already some evidence that type 1 interferon signaling had a role in neuronal homeostasis. Interferon beta is currently being used, one of the major therapies for another neurodegenerative disorder involving neuroinflammation, multiple sclerosis. It was also found to be essential for neuronal survival and outgrowth. And it seems to induce autophagy, which is that process of protein degradation and recycling. So without interferon beta, proteins seem to accumulate and also lead to neuronal death. But treatment with interferon beta appears to introduce or induce it rather, and allow this reduction of protein aggregates. It also seems required for these anti-inflammatory signaling of neurons, which, as mentioned earlier, if it's expressed by neurons, it seems to be able to convert those inflammatory cells into beneficial cells. But specifically, in clinical studies, it was seen that long-term type 1 interferon treatment of hepatitis C virus was associated with a reduced risk of developing Parkinson's disease. But also, most recently, it was shown that defective interferon beta IFNAR signaling was associated with dementia development in sporadic PD. So with that, I wanted to give an overview of what Parkinson's disease is. It's an aging-related neurodegenerative disorder that affects 7 to 10 million people worldwide. And the majority of these cases are considered sporadic or of unknown cause. So this means that no specific genetic defect or whatever seems to be causing this. Its hallmark symptoms are motor dysfunction, and eventually patients do develop dementia approximately 15 to 20 years after their diagnosis. The major symptoms they experience include tremor, loss of coordination, and muscle weakness. And of course, cognitive symptoms come delayed and after, and this includes memory loss, but also difficulties learning new things. And a slew of neuropsychiatric issues also plague PD patients, including anxiety and sleep disorders. And these disturbances actually, for some, may appear in a stage before motor symptoms develop. So this is an area of interest for clinicians and researchers to understand what processes occur before these motor and cognitive symptoms attack, basically. So the major pathological hallmarks, or at least the pathology in the brain that defines Parkinson's disease, includes the loss of these specific dopaminergic neurons in that area of the brain called the substantia nigra. So on the left here, you see the brainstem of a patient without Parkinson's disease, and this area of the brain is very marked with neurons that appear very pigmented, and that is the substantia nigra. But on the right, you see same region and brainstem from a Parkinson's disease brain, and that pigmented region is quite diminished. And this alludes to the fact that those neurons are actually not there, and very likely contributes to the motor dysfunction and lack of coordination that patients experience. But this neurodegeneration is also often accompanied by the presence of these spherical intracellular bodies called Lewy bodies. And these concentric inclusions include a protein called alpha-synuclein. And I'm going to be talking a lot about alpha-synuclein in the next few slides, so just keep that in mind. The cause of this neurodegeneration in sporadic PD is currently unknown. And there is unfortunately no cure or preventative treatment available. But there are theories on what is going on in PD. And this includes the involvement of alpha-synuclein. Now, there's a theory that the accumulation of alpha-synuclein and spreading results from actually initially an immune insult from the periphery, or rather something that enters the gut like a virus or a bacteria, that causes a misfolding of alpha-synuclein in the motor neurons that line the gut. And that misfolding pattern actually travels from the gut down the spinal cord and up to your brainstem, where the Lewy body inclusions would then start to form. And this is the basis of the Brack hypothesis, if you've heard of that before. Mhm. There. There's also a theory that these alpha-synuclein positive Lewy bodies are what's causing the neurodegeneration because they're toxic. And so the hypothesis is that there would be disease improvement if these Lewy bodies are moved or prevented somehow. And also that there's a lot of evidence showing that alpha-synuclein proteinopathy actually causes neuroinflammation, which then is hypothesized to exacerbate the neurodegeneration that's seen in PD. But there are some counterpoints to these theories that still make it unclear about the etiology of PD. This includes a weak correlation between alpha-synucleinopathy found in postmortem and the sites of neurodegeneration that are actually witnessed. There's also a pretty small but significant number of older adults that have Lewy bodies, but didn't present with any Parkinson's disease like symptoms. And this condition is called incidental Parkinson's disease, which there is a theory that these adults actually didn't, unfortunately, live long enough to then experience the motor symptoms that come from Parkinson's disease. Yeah, that remains to be resolved. And finally, it's not just alpha-synuclein that appears to be accumulating in PD. It's often the case that amyloid beta, which is actually more known in Alzheimer's disease, but also phosphorylated tau, also associated with Alzheimer's disease, are comorbid with Parkinson's disease, particularly in patients with Parkinson's disease dementia. Now, the clinical diagnoses of Parkinson's disease with dementia versus Alzheimer's disease is a bit tricky, and that's still being debated and discussed on a yearly basis by clinicians who are experts in this field. But the fact of the matter is we can't ignore that there are other pathologies that are occurring other than just simply alpha-synuclein accumulation. That begs the question, is alpha-synuclein driving Parkinson's disease pathology? So another feature of Parkinson's disease that's quite well established is immune dysregulation. Now, in the periphery, Parkinson's disease patients actually have higher levels of cytokines that induce inflammation, including TNF alpha, also interferon gamma, and this interleukin 1 beta or IL beta. Recently, it was also seen that peripheral T cells were found in the substantia nigra of PD brains, meaning that these T cells that are normally not found in high numbers in the brain might be entering somehow, perhaps because the blood-brain barrier is disrupted or what have you. But it suggests that there is an immune attack, or at least an immune response that could be contributing to neurodegeneration in Parkinson's disease in the area of the brain that's specific for that type of cell. And finally, inflammatory bowel disease patients or IBD patients who experience inflammation of the gut appear to confer a higher risk of developing Parkinson's disease. Now, these are all interesting because this implies that this peripheral hypothesis, this BRAC hypothesis of an peripheral insult can lead to neurodegeneration in the central nervous system. Now, within the brain, there's quite a bit of evidence suggesting that there is inflammation happening there as well, including activated microglia and active astrocytes in the area of the brain experiencing neurodegeneration in Parkinson's disease. But also there's quite a bit of experimental evidence and showing that increased pro-inflammatory cytokines in that area directly lead to dopaminergic cell death. So these are not far from understanding the true nature of the neurodegeneration. The background to what I studied for my PhD was that in 2015, Charet's group published that mice lacking interferon beta actually developed Parkinson's disease with dementia-like behavior and neuropathology. Now, this is important to note because mice don't naturally develop dementia or neurodegeneration like humans do. It usually needs to be induced somehow. And most models for Parkinson's disease was a direct overexpression of alpha-synuclein, for example, or a direct insult to the actual neurons residing in the substantia nigra. Now, this is a mouse that's just lacking an immune gene, and it seems to develop this specific neurodegeneration and neuropathological hallmarks that look like Parkinson's disease. So it was quite interesting to see. So this included, yes, this Lewy body-like inclusions that were alpha-synuclein positive, but also the behavior deficits were very similar to what you would expect the clinical progression to be in a human. So over time, these mice developed an inability to coordinate motor movement as well as learn. They were unable to learn memory tasks compared to wild-type mice. But they also exhibited this extensive dopaminergic specific neuronal loss over time. And that gave me a good background to study two aims. The first aim was to determine whether the absence of alpha-synuclein would actually affect the progression of this disease. Or in other words, I wanted to study, will a lack of alpha-synuclein actually improve the prognosis in these mice lacking interferon beta. And then my second aim was then, of course, to explore that context and origin theory, and to understand really the brain cell specific requirement of IFNAR1 in maintaining CNS homeostasis and also the natural behavior of mice. In other words, I wanted to look at whether or not IFNAR1 signaling differed between neurons and glia. And the expectation, or at least the hypothesis was, of course, they're going to differ, but how do they differ, and to what extent is what I was hoping to address. So for the first aim, this led to first study, and I'm happy to report that it has since been published. So you can go online and download it if you will. But the background of this is, of course, challenging the notion that alpha-synuclein is the primary driver of Parkinson's disease. And I'm not saying that it's not important, but I wanted to see it in an endogenous context. So alpha-synuclein, the protein, is considered a presynaptic protein localized at the presynaptic terminal, and it's encoded by the SNCA gene. Now, this gene is important because in familial PD, which is approximately 5 to 10% of the cases of Parkinson's disease, they can actually lead to the development of Parkinson's disease in that if the gene is multiplied, then the amount of protein actually increases by a lot. And that leads to aggregation, accumulation, and it seems to be what causes the dopaminergic neuronal death. There are also polymorphisms, meaning that this gene differs slightly in patients with sporadic Parkinson's disease. So it's not a direct genetic mutation, but these slight genetic differences could be contributing to the risk of developing Parkinson's disease. So of course it's of importance. And of course it's a primary component of these Lewy body-like structures, or that pathological hallmark. So this led to alpha-synuclein being a major target for developing compounds for treating Parkinson's disease. But again, as with science, it's always we want to be sure, and can we confirm this? Is aberrant alpha-synuclein the primary cause of neurodegeneration in sporadic PD, as other proteinopathies also seem to occur coincidentally in sporadic PD and PDD, but also that these alpha-synuclein overexpression models that I mentioned before they sometimes, or the success rate of recapitulating all the aspects of Parkinson's disease, including the motor dysfunction and dopaminergic specific neurodegeneration, varies depending on the way that the experiment had been designed. So my study aimed to address whether the lack of endogenous alpha-synuclein would actually affect the Parkinson's disease-like phenotype in these mice. So to do that, we developed a mouse that actually lacked both interferon beta and synuclein. And the genotypes that I looked at were to compare this double knockout mice, I'm going to call them a double knockout now, to wild type mice, interferon beta knockout mice, and also alpha-synuclein single knockout mice. And I took these mice and assessed them over time for their behavior and also the neuropathology that they develop. So this includes looking at their motor behavior, whether or not they could learn a process at different time points. But also assessed atypical behavior like anxiety and also pain sensitivity, which appear to have some involvement in Parkinson's patients as well. And I used a number of methods for this at different time points. But then I followed up these in vivo investigations by investigating immune signaling specifically in primary neurons that were derived from these mouse strains. So from the behavior studies, I found that these mice, with or without alpha-synuclein, if they lacked interferon beta, they actually experienced motor deficits over time. So by 6 months of age, both interferon beta knockout here in the dark gray bar and double knockout mice actually could not perform to the same extent as wild type and synuclein knockout mice. Now for learning, I actually found that not only did interferon beta knockout mice experience this deficit and inability to learn where to find a escape platform, but the double knockout and synuclein knockout mice were unable to find it over time. And this actually suggests that synuclein may have a very important functional role in neuronal, or at least in the memory formation process. Now, looking specifically at the dopaminergic neurons, there was a significant decrease in dopaminergic neurons without interferon beta, but also without alpha-synuclein. So this kind of confirms that alpha-synuclein is also important for the health of these dopaminergic neurons. But when we looked at the processes, it appeared that they were more significantly affected, or at least there were fewer processes in the mice lacking interferon beta with or without alpha-synuclein. And this was conferred by looking at the striatum that these processes were not as defined in the striatum of mice lacking interferon beta. Now, whether or not these Lewy body-like structures could form, interestingly enough, because of course the double knockout didn't have alpha-synuclein, we stained for phosphorylated tau, which has also been published to be detected in Lewy bodies from postmortem PD patients, but also amyloid beta, because we were interested in general of the co-expression of these molecules with or without alpha-synuclein. And we confirmed these morphological processes by electron microscopy, that both the double knockout and even the single synuclein knockout developed these concentric Lewy body shaped spherical inclusion bodies that didn't depend on alpha-synuclein expression. So this was quite interesting to us, very surprising, because of course, this was dependent on the, or at least this challenges a little bit the requirement of alpha-synuclein for Lewy body formation. But again, this is very descriptive at this point. So we also found that the lack of interferon beta results in accumulation of other proteins other than alpha-synuclein. Here you can see alpha-synuclein in dark. accumulating in the interferon beta knockout mouse. Of course, it's not detected in the double or single knockout mouse. But when we looked at amyloid beta as well as phosphorylated tau, in the middle columns here, you can see that the mice lacking interferon beta, with or without alpha-synuclein, also accumulated other proteins like alpha amyloid beta and phosphorylated tau. And we also detected extracellular amyloid beta plaques, like in absence of interferon beta, and these weren't detected in wild type or single synuclein knockout mice. Now, these protein aggregates were also associated with glial cells. So we stained here for, in the top panel is for phosphoalpha-synuclein. You can see a microglia labeled with a microglial marker, and it engulfing a alpha-synuclein positive aggregate in an interferon beta knockout mouse. So we took a similar investigation in the 4 genotypes mentioned before, and saw that only the interferon beta knockout and double knockout mice actually accumulated amyloid beta. And this is in the hippocampus. And this accumulation actually was associated with microglia and also astrocytes in interferon beta knockout. So then we asked whether or not neuroinflammation was actually induced because of the lack of interferon beta. And it does appear so, in that with loss of interferon beta, these mice lacking interferon beta, there was an accumulation of tumor necrosis factor alpha or TNF alpha, both at the expression level, but also at the protein level. And this was also associated with microglia. So there were more microglia expressing TNF alpha, and also astrocytes expressing the molecule MHC2, which would indicate that they are reactive in the interferon beta knockout brain. And when we looked at the double knockout and single synuclein knockout, there was an increase in TNF alpha positive microglia, both in the hippocampus and the substantia nigra. Interestingly enough, though increased microglial TNF alpha was seen, we actually also saw a strange increase in neuronal expression of TNF alpha in substantia nigra. And this is definitely a finding that was not expected. But in this panel here, you can see interferon beta and TNF alpha in red, and neuin, which is a marker of neurons, in green. Now, these neurons label both dopaminergic and non-dopaminergic neurons, and these TNF alpha positive neurons seem to be non-dopaminergic. So, I could go into the different neuronal subtypes in the substantia nigra that are not dopaminergic, but I will continue on. The point is that we detected neuronal expression of TNF alpha was of interest for us to continue our studies in primary neuron cultures. So just a short background on TNF alpha. It is also implicated in Parkinson's pathogenesis. And it appears that TNF alpha driven neuroinflammation could contribute to dopaminergic degeneration in PD. And this is backed up by some evidence of elevated TNF alpha expression, but also polymorphisms that confer a higher PD risk. And neuroprotection from amyloid beta induced toxicity requires active TNFR2 signaling, and interferon beta treatment seems to downregulate TNF alpha and MS. Now, I see here that I'm running out of time, so I will continue on. But basically, the cellular studies that we showed that in neuronal cultures, by treating the neurons with interferon beta or blocking the pro-apoptotic TNF alpha receptor, we saw a decrease in the amount of apoptosis that was actually higher in baseline in neurons lacking interferon beta. And this higher baseline was rescued, or at least the cell death was prevented by blocking TNFR1 or interferon beta. Now, the proteinopathy was also reduced in terms of amyloid beta and phosphorylated tau accumulation with recombinant interferon beta treatment. So to summarize, with interferon beta signaling, it could contribute to healthy aging, but with loss of interferon beta, it actually resulted in Parkinson's disease-like phenotype in mice that seem to be independent of alpha-synuclein. So quickly go through this. And then the second study, which I will only touch upon very quickly, is that for the second study, I actually looked at whether or not these effects could be cell-specific. What we know is that interferon beta has a lot of side effects in MS, and that, of course, all nucleated cells express IFNAR. So the effects of interferon beta that are different when regulating inflammation could be cell-specific. So we wanted to know what these cell-specific requirements were, and that study is in preparation. And I would love to talk more about it, but I think I need to skip over, or maybe not. I'm a bit lost with my cursor here, but sorry. Okay, yeah, sorry, I'm gonna slow down now. Let me just see. It's a little hard to navigate when I don't see any faces or hand waving or anything like this, but in any case, the premise of this second study was that we found that mice lacking IFNAR, or rather the receptor for interferon beta, also developed these Lewy body-like inclusions. And when looked at a cell-specific knockout, rather where mice lacked IFNAR in both neurons and astrocytes, they also seem to develop these Lewy body-like inclusions. So this led us to ask, does IFNAR loss also lead to this Parkinson's disease-like phenotype in mice? But also, is that effect specific to neuronal or astrocytic loss of IFNAR? Now, the methods were very similar to the previous study in that we took IFNAR1 knockout mice compared to wild type mice, but we also continued that study to or either neuronal specific IFNAR1 knockout mice or astrocytic IFNAR1 knockout mice. And we continued with unbiased molecular exploration using very cool tools, such as cell-specific transcriptomics using single nuclear RNA sequencing, which allowed us to look at, based on the genetic composition of the nuclei, which specific cells were differentially regulated upon IFNAR1 loss. And then we continued to look at the protein composition using state-of-the-art mass spectrometry, and also functional assays that using metabolic isotope labeling, we could differentiate the neuronal versus astrocyte energy consumption and also metabolic phenotype. So what we found was that global IFNAR1 loss actually did result, just like interferon beta knockout mice, in PDD-like behavior and pathological manifestations, and that it was actually neuronal IFNAR loss that contributed to the cardinal PDD phenotypes. We also found that astrocytic IFNAR1, not surprisingly, is also important for preventing other phenotypes, including the anxiety that we saw in these mice. So we then preliminarily concluded that neuronal and astrocytic IFNAR1 must be acting in concert to maintain CNS homeostasis. And we saw that IFNAR1 loss in general induces a lot of transcriptional changes that are quite different between different brain cell types, including neurons, astrocytes, microglia, oligodendrocytes. So it is quite important to note the cellular origin of IFNAR1 signaling. And also that proteome alterations and this differential energy metabolism actually occurs prior to cognitive decline in these mice. So these data sets can actually give us a window of opportunity to identify molecular targets that could potentially slow or reverse the disease process when targeted properly and specifically. So there's a nice figure with the interferon IFNAR signaling. You have functional neurotransmission, which leads to healthy aging behavior, but upon loss of IFNAR1, you have this protein accumulation like we saw in the interferon beta mouse. And this seems to be cell-specific in terms of the origin of behavior deficits. But this all seems to be stemmed from dysregulated excitatory neurotransmission confirmed by the functional assays. So in general, it appears that neuronal type interferon or type 1 interferon signaling is required for CNS homeostasis and the prevention of Parkinson's disease with dementia-like disease. So what we found was that disrupted interferon signaling was sufficient to cause Parkinson's disease with dementia-like manifestations in vivo, and that seemed to be independent of alpha-synuclein. And an important finding I found was that alpha-synuclein appears important for just regular cognition. So lowering alpha-synuclein strategies must be made with caution because it could result in detrimental effects to the neurons as well. And lack of neuronal TNF-alpha, like neuron-specific TNF-alpha and TNFR1 regulation may also be of interest to target. And that neuronal IFNAR1 loss, specific loss of IFNAR1 in neurons is sufficient to induce motor and cognitive phenotypes that resemble Parkinson's disease and dementia. Now, what we know is that IFNAR1 is required for energy regulation and mitochondrial function in CNS that was outside of my studies and was discovered independently. Disrupted energy homeostasis in study 2 was found to occur prior to cognitive decline. But in support, interferon beta has also been shown to regulate mitochondrial fission, and mitochondrial disruptions appear to occur upon inhibition of interferon beta IFNAR signaling in PDD, in human PDD. So what does that mean for PDD research, but also neurodegenerative disorders in general? So because we looked at other proteinopathies that appears to be of interest in, for example, Alzheimer's disease, induced neuronal interferon beta could have potential in targeting multiple pathologies by preventing protein aggregation, neuroinflammation, but also this defective brain energy metabolism that we saw in the study that unfortunately I could not go through in detail. But the overlap in pathology in Parkinson's disease with dementia and Alzheimer's disease could mean that targeting this dysfunction could benefit Alzheimer's disease patients as well. So in general, as with science in most studies, further study is warranted, specifically in identifying downstream cascades, because of course, IFNAR is such a broad target. Finding downstream pathways that actually lead to specific results could be beneficial in that if you target that, then you get rid of the unwanted side effects of non-neuronal IFNAR1 promotion, for example. If you were to promote IFNAR1 signaling in microglia, it could be very different and also in the opposite direction. But also we need to understand more about the baseline levels and also just in general, what the activation patterns are in neurons to understand interferon beta IFNAR1 signaling. And that requires further study. So with that, I have a lot of people to thank, including my previous PhD supervisor and co-supervisor, and also my co-authors from Diazazade and Avika's group, but also my collaborators in Germany, as well as here in Copenhagen, and the funding bodies Lundbeck and the Independent Research Fund Denmark. And thank you for your time and also your patience with me going back and forth between screens. But I'm very happy to answer any questions that you might have and give the floor to hopefully the panelists who will pop up again once I stop sharing my screen. Oh.
Yeah, Here we are,
on our.
magically appeared. So, yeah, thank you so much for your interesting and very detailed presentation. As I wrote in the chat, please feel free to type your questions, or if you raise your Zoom hand, then Dr. Young can empower you to actually speak. So while you are considering questions, I'll just gonna rephrase. Oh, Erica, you can read the one which.
Yeah. Thank you. Sorry, just a second. Stop share. Okay, perfect. Oh dear. After 2 years of using Zoom, it's still a lot to learn and unlearn and whatnot. So yes, thank you for your patience. Anyway.
Such a powerful technology. Yeah.
With great power, right? So how shall we connect?
Oh, okay. So I'll just read out. Dr. Vaughn is asking about dopaminergic neurons, that they are sensitive in Parkinson's disease, and the question is, do you think they're prone to this death, right, in Parkinson's? In Parkinson's is related with the expression of TNF alpha.
Yeah, I mean, it definitely could be. There's the selective neurodegeneration seen in Parkinson's is there are theories it's in relation to how dopaminergic neurons are actually so unique in terms of their morphology, because they have such long distances to communicate to and from. This leads them to being more sensitive to imbalances in their energy regulation, but also maybe it could be that their expression of TNF-R1, for example, the receptor for TNF alpha, could be highly dysregulated in that case. The exact mechanisms again are not really clear, but it could be that, let's say, if other neurons or if microglia are also highly expressing TNF alpha for other reasons, maybe peripheral inflammation or whatnot, that could be then targeting the TNF-R1 on dopaminergic neurons that could then lead to that cascade of specific neurodegeneration. But again, you know, what starts and what's being caused by it, it's hard to know.
Thank you so much. So I'm also I'm curious about interferon beta, because from your talk it seems such a great molecular and so forth. I think we have like a recent data set where we added interferon to microglia, happily killed neurons.
Yep.
Does that surprise you, or can you provide us with some insight, like how this molecule and under what circumstance it can actually be not so beneficial?
So yeah, for sure, because I mean, in our own data set where we looked at, and unfortunately I wasn't able to show the results from the single nuclear RNA-seq, but the microglia specific population without interferon beta actually had this reduction in phagocytic activity. So it could be that when you add interferon beta to microglia, it increases their ability to phagocytose, or maybe it promotes that inflammatory phenotype, for example, or when it's actually processing what occurs in pathogenesis. So I'm not surprised that it would then contribute to neurotoxicity in that case, because it could be that interferon beta signaling with neurons is local, for example. So it would rely on interferon expressed from other neurons, or yeah, I think the context really matters, and it doesn't surprise me that microglia would be reacting differently.
Okay, yeah, Dr. Rowe.
So I'll ask my question orally because it's a little convoluted. But before I ask my question, I just want to say congratulations on the successful completion of your PhD. I haven't had a chance to say that to you. So Dr. Villanueva, thank you very much. That was a wonderful talk. At the beginning of your talk, you made the comment about the use of, I'm always confused with this signaling molecule and receptor and the use of origin and context. And they say, you know, the origin of the signaling molecule can matter. And classically, when you think about that, I question that because I can understand context of the downstream, you know, whether there be other signaling molecules in maybe in neurons, DAMPs or PAMPs are out there and affecting the response of the cell plus the whole milieu of the cell. But the question about origin, how can something which is classified as a signaling molecule from two different sources be interpreted differently by the same receptor? And the question I have is, can you comment on that? And are they actually the same molecule? Should we be calling them, for instance, if it's IFNAR as the receptor and it's interferon beta is the signaling molecule, are they really IFN or is it interferon beta 1A from the same two sources, or are they actually different molecules?
Yeah, I mean, definitely isoform differences. I mean, we're assuming that there are only so many isoforms, and interferon beta does have at least 2 isoforms that I know of that are used in the clinic. But it could be that at the very least with other inflammatory cytokines in the neuronal context, it seems to also involve some co-expressed surface markers that if, let's say, a glial cell secretes interferon beta as it would to recruit other immune molecules, it could be standalone. Whereas the interferon beta expressed by neurons could be co-expressed by another molecule that's within a smaller space. So the context, or the localization of it also could play a role in how that specifically different signaling cascade occurs. And I'm not sure if that answers.
Yeah, I think that kind of what I'm questioning is it that molecule, or is it a complex set of protein molecules that are actually acting as the signal?
I mean, I wouldn't be surprised if also the downstream signaling complexes that are in neurons, the composition of the complex that occurs when IFNAR is bound could be a little bit different from what it would be in a glial cell, for example.
Okay.
Interesting. So
yeah. I've always struggled with this, honestly, when I see these things, and I've never really understood the idea of how origin matters unless someone said, okay, these are actually slightly different proteins or something like that.
Yep,
but signaling molecules, yeah.
But I mean, proteins, like, to be honest, with internalization, all this, these mega complexes that can occur when signaling happens. Just looking at the death receptor complex, for example, that when you agonize TNFR1, there are so many different types of proteins that can be recruited to that complex that can actually exact different cascades that I'm also like, okay, wait, how and when do these specific ones become activated? But I think that's still, that it's so, there's so many different variations that can happen that it's not surprising that we don't know enough about what actually occurs after binding. And I think it is still technically challenging to investigate those specificities, if that makes any sense.
Yeah, no, thank you very much.
Neurons is not a picnic, so.
Thank you, that's wonderful.
Yeah, thank you. So before Shirley, I'll ask a question from your former colleague, Jocelyn, who left Edmonton. So she's asking whether you have considered glycans and sugars on these molecules in terms of their effect and origin also.
No, and that is a very interesting point, because that could also for sure affect it to a different maybe area of the cell surface, or different cells based on their cellular or membrane composition, for sure. So I mean, that area of resolution, or that I would love to see happen, but that was unfortunately not in the scope of my my research.
Okay, thank you. Shirley, please ask.
Thank you so much for the fascinating talk. I want to ask a question about your science journey. Did you ever have any moments that made you doubt whether you wanted to continue a career in biology or in neuroscience? How did you convince yourself to continue doing research in the academic field?
Yeah, so first and simple answer is constantly questioning it. And the second is, well, when I finished my MSc and was wondering if I should do a PhD, I got some very good advice from Andis, saying, you know, it is good to know when, if you want to take on something like a PhD, then, you know, you should have some degree of certainty. So I actually went to Vancouver and took a job in a research lab that was very similar, but also like to learn different techniques about. And I thought I would be happy being a technician for, you know, but as the years progressed, and of course I was in a lab where my immediate supervisor was also very keen on involving me in the process of experimental design and all this, it rekindled that interest to, okay, well, then you can take charge of your own research. You just have to, you know, go through the motions and pursue something that seems scary, but in the end, you know, I think if once you know that's what you want, then you go for it and take the opportunities as they come. Also seek new opportunities, because that's being proactive is also part of the process. I'm not sure if that answered your question, but yeah. It's,
Yeah, I myself had a similar question. So you are in this unique, you have these unique experiences of working in three very distinct places on this planet, meaning Okanagan, Lower and Copenhagen. So can you comment on how research and life compares?
well, I mean, to be honest, Kelowna was so relaxed. In the lab, it was not relaxed. No, I'm just kidding. I mean, I liked how effective and a small group could be. And also the teamwork that can happen in a smaller group, because when you get to a bigger lab, it's a little bit difficult to find your place in you also have subgroups, of course, and you can work really well with that subgroup, but in the grand scheme of things, it's sometimes a bit difficult to communicate with the other parties, but also coordinate your research in that sense. I mean, Vancouver was also very high paced, and I mean, I loved my time there, but it was also a very intense part of my career, in the sense that I wanted to contribute more. It was very motivating. And of course, that's why I jumped onto doing my PhD elsewhere. But I would say that it comes with a lot of pressure. And I guess the key is just not to lose yourself in that, because of course, well-being is very important and balance is also very important. So when I came to Copenhagen and people actually did leave their desks at 4 or 4:30, maybe even earlier when you have kids, that actually opened up, okay, well, this high intense pace of research can still occur with having some, you know, semblance of a life outside of it. So it was really nice to have the experience in Kelowna, where it was very conducive to learning, but also having that step up to I would say, you know, learning about the non-science aspect of science, if that makes any sense. Academia can be quite harsh, but it doesn't have to be. So I guess that's the take-home message, and it can be difficult and can take time to find that. Speaking from someone who's still looking, in any case.
Can I ask a question?
Yes, of course.
You started this all by saying you were a very proud first-generation Filipino Canadian, which is a wonderful way to introduce yourself. How do you feel that has played into your various experiences?
Well, I think it's definitely created a more humble mindset. And I say that because I know the opportunities afforded to my parents, for example, were very difficult. But I also recognize that I think I'm in a very privileged position compared to a lot of people of my background. So I wanted to recognize that, acknowledge that, my parents worked hard to get me to a point where I was freely able to attend university and also to pursue these different options. And I also recognize that that's not easy. And I'm not saying that, like everybody can end up here just for fun, because I feel like I've already had that extra level of support that isn't definitely not available to everybody. So with that, I thank my parents, who I think are part of this as well. But in any case, I wanted to provide a voice for, I know at least 2 other Filipina researchers here in Copenhagen as well, which is a very small community, but it is nice to see, despite our different baselines, we were able to arrive at a similar position. And again, not easy, but very commendable on my colleagues' parts. So.
Awesome. That's so great. I was going to ask you, actually, I don't know enough about the diversity in Denmark to know whether you would have found many other.
It's definitely, I mean, there are many different communities here who emigrated from other parts of Europe, but the composition is definitely different from even Toronto or like any part of North America that you would imagine. So the dynamic is a bit different. The way they approach immigration is also a little bit different. But again, recognizing that it is a different country. So it's a little bit like being in a parallel universe. But
thanks so much.
No worries.
Okay, anybody, any other questions? So shall we wrap this up? What do you think, Robin? Yeah. Okay. Well, in that case, yeah, I just gonna thank again our speaker, Dr. Villanueva. Thank you so much for being with us and sharing all your research and experiences. And thank you to the funders and organizers, of course, for making this event possible. So thank you so much, and thank you for attending.
Yeah, thanks a lot. So do we stop? How do we do this?
Yeah, I'll stop the recording. Sorry, I've lost the button. Stop recording. There we go.